Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good morning to all the examiners. My name is Nur Fadzrin binti Arifan. Today I would like to present about my final year project progress entitled Study on Biological Control Approach Against Bipolaris cactivora, the causal agent for fruit rot disease of Heliocereus species. My presentation is containing the introduction, problem statement, significance of study, objective, materials and methods, result, references, and lastly, Gantt chart. The common name of Heliocereus species is dragon fruit, Heliocereus undatus, Howard, Britton, and Roos. The red skin and the white flesh. The fungus that is responsible causing the fruit rot disease in dragon fruit is Bipolaris cactivora, also known as Bipolaris cactivora. Fruit rot is the most damaging disease in dragon fruit production. Bipolaris cactivora can survive in different cactus varieties with the main source of primary inoculum. Affect several parts in dragon fruit plant. You can see on the picture beside here is the symptom of fruit rot disease on Heliocereus species. The picture is taken by Earn et al. 2015. The main problem here is the usage of chemical fungicide as main killing agent against the fruit rot disease. Most of the farmers choose chemical fungicide. It is because the price of chemical fungicide is much more lower than the organic fungicide. The overusage of chemical fungicide will create the fungicide resistance, the toxicity towards non-target organism, and also unfriendly to environment. The way to overcome the effects of the chemical fungicide is by applying the biological fungicide to control the disease outbreak. By conducting this study, it is possible to identify new potential biological control agent as a biofungicide that is safer for the environment. This study containing three main objectives, which are to obtain a pure culture of Bipolaris cactivora of dragon fruit rot disease. Number 2 is to determine the optimum environmental factors influencing the growth of Bipolaris cactivora, to monitor the activity of plant extraction and antagonistic fungus and bacteria against the Bipolaris cactivora. The materials that we use in this study are 10% of Clorox, 70% of ethanol, PDA, NA, V8 juice agar, hydrochloric acid, sodium hydroxide. First step is isolation and identification of Bipolaris cactivora. The environmental factors influence growth of Bipolaris cactivora. The screen antagonistic activity of fungus and bacteria against the Bipolaris cactivora. Poisoning agar test of various plant extracts against Bipolaris cactivora. Lastly is data analysis. Okay, we go to the experiment number 1. Starting from sampling dragon fruit rot fungus, then we went to primary isolation from host tissue. The next step is phenotypic identification, and lastly, the phytogenicity test. This is the result from experiment 1. Phenotypic identification of fungi. On the left side is the upper side colony culture of Bipolaris cactivora from primary isolation. Meanwhile, on the right side is the lower side colony culture. The colony was suspected to be a colony of Bipolaris species based on their growth characteristic and physical appearances. Conidia pores were pale to light brown with a ring of diurnal zonation of dark brown to black on the lower side of it. This finding is quite similar with the research done by He et al. 2012. Besides comparing the colony physical appearances with the previous colonies stated in research paper, the phenotypic identification of the fungal has been done to examine the fungal hyphae and spores by using lactophenyl cotton blue as a dye. The identification reconfirmed that the fungus is Bipolaris cactivora. Several main characteristics of fungal phenotypes were used to identify and recognize the fungus. As we can see in the figure 3, this is the spore of Bipolaris cactivora under 40 times magnification. And figure 4 is spore and hyphae of Bipolaris cactivora under 40 times magnification. The Bipolaris cactivora have hyaline hyphae. Conidia pores are light to dark brown, mostly curved or canoe shaped. Okay, for experiment 1.2, this is the result of the coach postulate after 3 days of inoculation. Both symptoms show higher similarity in type of injury and the physical appearance. It is small, light brown spots were initially observed on the surface of the fruit. These symptoms quite same on the previous research by He et al. 2012. For experiment number 2, there are 4 environmental factors that we used in this experiment. This is the result for experiment 2.1. There is no significant difference in the mean radial growth of Bipolaris cactivora. Value of P is higher than 0.05 according to one-way ANOVA test. The fungus on PTA media show the larger radial growth, and in media, the fungus cannot grow well because it does not consist specific ingredient required for the fungal growth. Next, the effect of different temperature on radial growth of fungi in centimeter. There is a significant difference with p-value is lower than 0.05. So from the result, we can conclude that the best temperature for Bipolaris cactivora to grow at maximum rate is 25 degrees Celsius, followed by 30 degrees Celsius. This result is aligned with research by year 2015. Okay, next we move to experiment 2.3. There is no significant difference in the mean radial growth of Bipolaris cactivora because the value of P is higher than 0.05. The pathogenic fungus can grow until 4.49 cm at the media with pH 6, but it can reach only 4.28 cm at the media with pH 5. Okay, for experiment 2.4, there is no significant difference in the mean radial growth of Bipolaris cactivora because the value of P is higher than 0.05. The figure shows that Bipolaris cactivora that has been exposed to 24 hours light achieved the maximum radial growth by 5.11 cm, followed by 12 hours light plus 12 hours dark. Then we move to the next slide. In this experiment, we use 2 different methods, which are dual culture assay and double click. This is the result for experiment 3.1.1. As we can see, there is a significant difference in the mean radial growth of Bipolaris cactivora. The value of P is lower than 0.05 according to one-way ANOVA test. Based on the graph, they successfully inhibit the growth of the fungus by 50% compared to the control. Move to experiment 3.1.2. There is a significant difference in the mean radial growth of Bipolaris cactivora with the value of P is lower than 0.05 according to one-way ANOVA test. There are 6 Bacillus species able to inhibit the growth of this pathogenic fungus. However, the rest 3 Bacillus species show the lowest percentage of inhibition. For experiment 3.2, there is a significant difference in the mean radial growth of Bipolaris cactivora with the value of P is lower than 0.05 according to one-way ANOVA test. There are 6 Bacillus species able to inhibit the growth of Bipolaris cactivora. However, the rest 3 Bacillus species show the lowest percentage of inhibition. Experiment number 4. Poisoning agar test of various plant extracts against Bipolaris cactivora. There is a significant difference in the mean radial growth of Bipolaris cactivora with the value of P is lower than 0.05 according to one-way ANOVA test. Clove is the best antifungal against Bipolaris cactivora. For path forward session, I already have 3 more assessments that I need to complete in order to finish up my final year project research. The 3 assessments are data analysis, thesis writing, and article journal writing. For your information, this experiment is designed by using the complete randomized design. The data analysis is analyzed by using analysis of variance or ANOVA. The significance of treatments determined by Tukey's HSD test. These are all the references that I use as a guidance. Based on the Gantt chart here, on this June, I supposed doing my research poster and article journal writing. Submission of the thesis, research poster, and article journal will be expected on July. That's all for my final year project progress presentation. Thank you for lending me your ears and time. I wish you have a good day and Assalamualaikum.